Hi, welcome to my narrated lecture for Chapter 1, Exploring the Microbial World. So first, what are microbes? A microorganism is just an organism that is too small to see with your eye, so you have to see it with a microscope. Most microorganisms are unicellular, of course, but there actually are some that are multicellular. Most of them are prokaryotic. You may remember from general biology that a prokaryote is an organism that doesn't have a nucleus in its cells. Prokaryotes are always unicellular organisms. Most microbes are prokaryotes, but you also have some that are eukaryotes that do have a nucleus. So you have microbes just everywhere, absolutely everywhere, in every environment on Earth that can support life at all. And they always live in communities with each other. So you'll have multiple different species of microbes living together and doing different things for each other in basically symbiotic relationships. So you have microbes that can live even in places where other life forms can't survive. Those are called extremophiles. So an extremophile is a type of microbe that loves harsh environments. Could be places that are very cold, places that are very hot, places that have a lot of pressure, like the bottom of the ocean, places that are very acidic or very salty or very basic. So all types of places that, like, you or I could not survive, an extremophile, some type or other of extremophile, can frequently survive there and would be some type of microbe. So since they can live in so many different places on the planet, and since they're so small, there's far more microbes alive today than any other type of life, like by far. So they're the most abundant life form or type of life form on Earth, both in terms of the number of individual organisms that are alive and in terms of the number of different species that exist today and have existed in the past. They're very important. They're important for the environment, so they're important for us, for agriculture, because, you know, the environment is important for us. They're important for industry and technology. There's a lot of uses for microbes in biology and in other industries. They're important for our health. They cause disease, some of them, only a few of them cause disease actually, but some of them cause disease, and also some of them help us by living basically in symbiotic relationships with us. They impact every ecosystem on the planet because they live in every ecosystem on the planet, and they're required for the health of ecosystems. And there's a special subfield called microbial ecology that studies the impact that microbes have on the ecosystems where they live and how they are important in maintaining the health of those ecosystems. So microbes are either prokaryotes or eukaryotes. A prokaryote is a cell that does not have a nucleus. It's actually defined as not having a nucleus. Prokaryote literally means before a nucleus. And the nucleus is where a cell would normally be keeping its DNA or its genome, all of the DNA that it has, or all of the genetic information that it has. So in a prokaryote, since there's no nucleus, they can't store it there. Instead, they have what's called a nucleoid, which is just kind of a poorly organized part of the cell where their genome, where their DNA is all kind of bunched up. Then they can also have plasmids, which would be little circles of DNA that are separate from the nucleoid. And the genome is just all of the genetic information in the cell. For most cells, that means all of the DNA in the cell. Well, actually, I should say for cells, that means all of the DNA in the cell. For viruses, it sometimes means something different. Eukaryotes, in contrast, have a nucleus where they keep their DNA genome. And then they have other organelles around the nucleus that are closed in with membranes. And in chapter 2, we'll go through all these other organelles. You probably remember a lot of them from general biology. We'll go through all the structure of these prokaryotic cells and eukaryotic cells in chapter 2. Some of the activities that microbes have are metabolism, growth, and evolution. So all living things have these activities actually. Metabolism is just chemical reactions that occur inside the cell or that are carried out by an organism. So all of the chemical reactions that an organism does are together that organism's metabolism. The basic purpose of metabolism is to get energy for the cell or for the organism, and then also to build structures, to use that energy to build structures in the cell so the cell can grow or the organism can grow, and ultimately so that that organism can reproduce or divide. In order to have a functioning metabolism, a cell has to have enzymes, which are protein catalysts. A catalyst, as you may remember, is something that speeds up a chemical reaction but is not changed by that reaction. So it's not a product, it's not a reactant, but when the catalyst is there, the reaction goes faster. If you have a catalyst that's a protein, that's an enzyme. So a lot of these reactions just wouldn't be able to happen fast enough if you didn't have enzymes to make them go faster. One major part of metabolism is protein synthesis. The process by which the genome is used to make all the proteins in the cell. You have two major processes within protein synthesis, which we'll go into in more detail in a later chapter, and which you probably also remember from general biology. Those are transcription and translation. Transcription is using the DNA genome to make RNA, or mRNA specifically, messenger RNA. That process is carried out by the enzyme RNA polymerase. And then you also have translation, which is the next process, the process of taking that mRNA and using its information to build a protein, and that's carried out by the ribosome. Another important part of metabolism is DNA replication, which is copying the genome, making another copy of the genome so that the cell can divide. Growth, just for a larger organism, for a regular organism, growth means like the whole organism getting bigger, so making more cells so the organism gets bigger. For unicellular organisms, it can't really mean that, of course, because one cell is already the whole organism. So for unicellular life forms, growth means that the cell itself gets bigger until it's able to divide to make two cells. Then if you're talking about a population of microbes, then growth means how quickly the cells in that population are dividing so that the whole population gets bigger. But those cells are not together as one organism. They're independent individuals all living together. So that's a population. These microbes would be reproducing for the most part asexually, of course, so that each new cell is just a copy of the parent cell that made it. Microbes undergo evolution, as do all living things, which means that they change over time, and that process is driven and shaped by natural selection. So in evolution, first you have mutation, changes to the genome, random changes to the genome. Those changes create new alleles. As you remember from intro biology, an allele is a version of a gene. So for instance, if you have a gene for eye color, you could have brown version, green version, blue version. These will all be different alleles. You could have a brown allele, a green allele, a blue allele for that gene. The actual sequence of nucleotides in each of those alleles would be slightly different. So mutation is how you get new alleles, and then those alleles may be beneficial, or they may be harmful, or they may be neutral. Depending on how they affect that organism that has that new allele, it may be more likely to be passed on or less likely to be passed on according to natural selection. So over time, you're going to see the frequency of alleles in the population change, which would mean how many individuals in the population has that specific allele. That number is going to change over time, driven by natural selection. If you have a beneficial allele, you're probably going to have more individuals that have the allele as time goes by. If it's a harmful allele, then probably as time goes by, less individuals will have it. And so the population will change in that way, and that's evolution. Microbes have basically accelerated evolution. They evolve much faster than any other type of life. And there's multiple reasons why that is. One of the main reasons is just that they are so small and they divide and reproduce so quickly. So they have a very fast generation time. These are some more activities that some microbes have. So not every microbe is able to do all of these activities. Different microbes can do different subsets of these, or some are all or none even. So a lot of microbes are able to move, they have motility, but not all. And there's a variety of different structures that can let a microbe move. We'll go over a lot of them in the next chapter, actually. So some microbes can move and some cannot. Then you have differentiation, which would mean just changing the shape or the features of the cell. So some microbes have kind of different forms that their cell can take. A lot of microbes actually do not have the capability to differentiate into a different type of cell, but some can. An example would be the endospore, which we'll also talk more about in chapter 2. An endospore is just a really hardy form of a bacterial cell. So some bacteria, for instance, the bacteria that causes anthrax, if conditions get too harsh or there's not enough food around, it can change its shape to become an endospore. Which is very tough. It can survive all types of conditions. So that's a type of differentiation. Then you have intracellular communication. Actually, a whole lot of microbes, most microbes, can do some type of intracellular communication. That would be communication between different cells. So microbes can communicate to other microbes that are around them, either of the same species or even of a different species that they're living in community with. So the microbe would generally, there's different ways that this can happen, but in general, the microbe would release some type of chemical that is going to carry that message to other microbes in the local environment, and those microbes will have a receptor that can bind that chemical message. Then you have finally horizontal gene transfer, which means transferring DNA between individuals. Not all microbes can do this, only some. And there's actually multiple different ways that you can do this. We'll talk about the specific ways that you can do horizontal gene transfer later in the semester. But in horizontal gene transfer, you have one individual giving a gene or giving several genes to another individual that's near it. Could be the same species, could even be a different species. So microbes can exchange genes with each other this way. Usually, specifically, it's some type of gene that's on a plasmid. So usually when they're doing this horizontal gene transfer, what they're actually doing is sharing plasmids with each other. Whatever genes are on that plasmid are going to get shared. Horizontal gene transfer ends up being really important in medicine because it's a way that antibiotic resistance can spread. So if you have one microbe that has a mutation that gives it an allele, a new allele of a gene that makes it resistant to an antibiotic, if that gene if that new allele that gives resistance is on a plasmid, then a lot of the time that microbe would be able to share it with other microbes around it. And so resistance to the antibiotic can spread very quickly that way, even between species in some cases. So microbes have been very important in the history of the Earth and life on Earth because microbes have had a huge impact on the biosphere, which is the part of the Earth that can support life. So Earth itself is estimated to have formed 4.6 billion years ago. And when it first formed, there was no oxygen in the atmosphere, which means that the Earth could not support life forms like us. It would be impossible for us to live there with no oxygen in the air. Not too long after Earth first formed, the first microbes emerged. Would have been, we are estimating, between 3.8 and 4.3 billion years ago that the first microbes came to be. And they would have been the first life form. So the first life form on Earth was a microbe. It was some form of unicellular prokaryote. And the name we give to that life form is LUCA. LUCA stands for Last Universal Common Ancestor. So LUCA would have been the organism from which all life on Earth today is descended. So every organism has LUCA as an ancestor, as its earliest ancestor, basically. The process of how LUCA came to be, or how life arose, that we do not know. That's an area of active research. Not too long after life emerged, you had a major development, something major evolved, which was photosynthesis. So about 3.6 billion years ago, the first microbes evolved that could do or could perform photosynthesis, which of course is just the process of using light for energy. So photosynthetic organisms are able to capture energy from light and use that to build the molecules that they need. In general, when you think about photosynthesis, you think about a plant breathing in carbon dioxide, breathing out oxygen. But of course, the first photosynthetic organisms were microbes, not plants. And also, when photosynthesis first evolved, it did not produce oxygen yet. So it was like a different type of photosynthesis than what we're used to. But it was still capturing light for energy, which makes it photosynthesis. Then about 2.6 billion years ago, an important group of bacteria evolved called the cyanobacteria. The cyanobacteria are photosynthetic, and they do the type of photosynthesis that produces oxygen. So they started mass producing oxygen, basically, and pumping it out from their little cell bodies into the environment. Once the cyanobacteria emerged, the concentration of oxygen in the atmosphere slowly began rising over almost a billion years. It took a very long time for the oxygen concentration to rise in the atmosphere, but it did start rising very, very slowly until you eventually wound up with the atmosphere that we have today that's about 20% oxygen. So it would be impossible for us to exist if these cyanobacteria had not evolved so long ago and started producing oxygen in enough quantities to actually change the percentage of oxygen in the whole atmosphere for billions of years to come, which is pretty impressive. Right, so these cyanobacteria are basically, they're what allowed us to exist. They are what allowed evolution to continue to create more complicated life forms. Oxygen is, of course, something that we use for energy. It's very efficient. We're going to learn about the process that we specifically use to get energy that requires oxygen later on in the semester, and you may remember it from general biology too. But there's lots of different ways to get energy. If you're using oxygen to do it, it's very efficient and you can make a lot of energy compared to other processes that you could use instead. So in order for something really big like you and me to evolve, we need a lot of energy. We need to be able to use oxygen to make that energy. Continuing about 2 billion years ago, you had the first eukaryotes evolve. They were, of course, still microbes. And about 1.4 billion years ago, the first multicellular life forms evolved. But it still wasn't things like us, it was still microbes. So Algae is actually a type of microbe. It's a multicellular microbe. It's like the most common multicellular microbe. So I'm sure you guys have seen algae, growing in a lake or a pond or something. That's actually a type of microbe, and that would be like one of the first types of multicellular life. Types of life that we're more familiar with, things that look like us, are much more recent. So large multicellular life forms didn't evolve until just about 500 million years ago, which is very recent in terms of the whole history of the Earth. So that's when you would have shellfish and plants and things like that, and insects start to evolve barely 500 million years ago. Which means that microbes have been the only type of life on the planet for the majority of the history of the planet, and they're the most abundant type of life on the planet. So really, life on Earth is, in some sense, mostly microbial. Well, actually, in many senses, mostly microbial. Life is divided into 3 sort of major groupings called domains. So in sort of older classification systems, you have like the kingdoms of life, like there's the animal kingdom and the plant kingdom that you're probably familiar with. Today we don't use those systems anymore, although our modern classification systems are influenced by that old system. But today we use domains as our biggest grouping of species. And you have 3 major domains of life, into which all life forms can be organized. 2 of them are microbial. Bacteria and archaea are entirely microbial domains. So the bacteria are prokaryotes. They're the oldest life forms, so they're the first to evolve. Then archaea are also prokaryotes, but they evolved more recently. All microbes again. Then you have eukarya, which is the most recent domain to evolve. Most of the life forms that you think about when you think about, living things would be eukaryotes, or belonging to domain eukarya. So all of your multicellular life forms are in eukarya, all your big stuff, and also some eukaryotic microbes as well. Archaea is newer than bacteria, and then eukarya is the newest domain of all. So archaea and eukarya are actually more closely related to each other than eukarya is to bacteria. So we're closer to eukaryotes than we are to bacteria, evolutionarily speaking. So in this little diagram of the tree of life, it's very simplified, but at the very root you have Luca, the last universal common ancestor, which would have been the first or one of the first organisms to evolve. And then from Luca you get the bacterial domain. So in early Earth's history it was all bacteria. Then at some point archaea would have evolved from that. Archaea are also microbes. And then very recently, comparatively, eukaryotes evolved, or eukarya evolved. And you can see that archaea and eukarya branch more recently from each other, whereas bacteria branches from eukarya way back at Luca. So we're closer to archaea than we are to bacteria. So microbes have a big impact on human health, of course. Most pathogens are microbes. A pathogen is just something that causes disease, but since mostly they are microbes, you could also just think of a pathogen as being a microbe that causes disease. In the whole scheme of all the microbial life on Earth, very, very few microbes cause disease in people. Most of them are not harmful to us at all, but some of them cause disease. Once upon a time, pathogens or diseases caused by pathogens were the leading cause of death. So in this graph here, you can see that around 1900, the leading causes of death were the flu and pneumonia. Then you had tuberculosis. Then you had gastroenteritis, which is an intestinal disease. These conditions are all caused by pathogenic microbes. So microbes are killing people more than any other thing back at the turn of the century. Since then, we have developed a lot of stuff. We've developed sanitation, like hand washing and stuff. They didn't used to do that in hospitals because they didn't know it was important. Once they developed that, it started saving a lot of lives. In addition, we have antimicrobial drugs now, and we have vaccines. So between those things, we have reduced the number of people that die of pathogens by a huge amount. So today, our leading causes of death are heart disease and cancer, not pathogenic diseases. So that's the negative impact of microbes on your health, but there's also positive impacts that different microbes have. So you guys may have heard of the microbiome, or perhaps not, but the microbiome is the community of microbes that lives on and in your body. So on your all of your skin, it's full of microbes, you guys. There's little things living all over you, and all throughout your gut, in your mouth, in your intestines, your tummy, all of it is full of microbes. Any place that has any kind of communication with the outside world, microbes are going to come in there and they're going to start living. So that might sound kind of creepy, but it's a good thing actually. The microbiome is very important for your health. Most of the species that are living in your microbiome are not harmful to you and cannot be harmful to you. They're just going about their business basically. Some of them that are living inside of your gut can actually help you digest your food, so they benefit you in that way. The ones in your gut will also have a major role in developing your immune system and helping your immune system learn the difference between something that's foreign and something that belongs to you, so that you don't have your immune system like overreacting or attacking your own tissues. So that's really important. Also, the microbiome just kind of helps keep out like bad microbes or pathogens. So if you have a pathogen come along and try to start living, say, in your intestines, that could cause gastroenteritis, it's going to have to compete with all the other microbes that are already living there. And those microbes have like well-established populations, and it's like their home turf. So they have an advantage in that competition, and the new microbe may not be able to establish itself, and if it can't establish itself, it can't cause disease. So the microbiome is real good for you. Microbes are also really important for agriculture, because microbes are kind of, in a way, controlling the way that different nutrients move through the environment. So you have things called nutrient cycles, which are the movement of these essential nutrients throughout the environment, through the soil, through the water, through the air, and the different forms that those nutrients take. And it's microbes that are basically facilitating that, so that nutrients can cycle through the ecosystem as necessary. So in order to have like good soil that has a lot of nutrients that can support plant growth, you have to have microbes to make that possible. You have to have nitrogen. That's one of the things that plants need to grow. Nitrogen is very abundant in the atmosphere, but plants can't use it in that form when it's in the atmosphere. You have to have microbes that take it from the atmosphere. When it gets into the soil, they convert it to a form that plants can use. So they're cycling that nitrogen from the air into the soil so it can go into the plants. Microbes, as they're cycling nutrients through the environment, they're also converting the nutrients to different forms chemically. So nitrogen that's in the air is in the form of N2, or molecular nitrogen. Nitrogen in the soil is going to be in a different format. It might be in ammonia, it might be in nitrate, different things. That conversion of N2 from the air to ammonia or nitrate in the soil, that's carried out by microbes. And that's called fixation. Anytime that a microbe is taking something from one form to another, if the first form is not useful to other life forms and the new form is, that's fixation. So plants cannot use N2. It's useless for them. They can't interact with it at all. But they need nitrogen. They can use nitrogen that's in the form of ammonia or in the form of nitrate. depending on what type of plant it is. And so it's microbes that are taking that unusable N2 and putting it into a usable format for the plant. So that's called nitrogen fixation. So different nutrients are going to be fixed by different microbes and made available for plants and other organisms to use. Right, that means they're very important. You can't have nutrients provided to plants if you don't have microbes. And if you have good soil with all those microbes in it that are doing all that nutrient cycling and fixation, then you don't need to use so much fertilizer, which is awesome because it's cheaper for you and it's better for the environment. One example of this kind of relationship between microbes and plants would be legumes, which are bean plants. They have these little balls on their roots. And these balls have bacteria living in them that are nitrogen-fixing bacteria. So they're able to take nitrogen N2 from the atmosphere and convert it to ammonia, which can be used by the bean plant. So you would have N2 kind of penetrating into the soil all around here, around the root system. The plant can't use that. But the nitrogen is going to go into these little balls, and the bacteria that live in there will turn it into ammonia, and then the roots will take that ammonia up and the plant will use it. So that's the importance of microbes for plants that you're growing, but it's also important for livestock too, because they need microbes in their microbiome to help them digest their food and to support their own immune systems as well, just like you do. But they actually, a lot of animals have a huge reliance on microbes for digesting their food, specifically animals that eat grass, which are called ruminants. That would be most of our large cows. That's cows. Also horses, deer, but for agriculture purposes, cows. So those animals eat grass, but they cannot digest grass. Grass has a lot of cellulose in it, which is a long polysaccharide. It's made out of, a polysaccharide is just a giant carbohydrate, basically, a giant chain of sugars. And the specific sugar that cellulose is made out of is glucose, which is, good eating. It's good for nutrition. But cows can't actually use it when it's in when it's in cellulose. They cannot break the cellulose down to get those little glucoses out of it. There's no way for them to do it. No animal actually is able to break down cellulose to get the glucoses out. We just don't have the right enzymes that you need to do it. But some microbes do have those enzymes. So inside the stomach of the cow, you have microbes living that are able to digest that cellulose, take it into little glucose molecules, and then the cow's gut will absorb those glucose molecules and it gives the cow fuel. So if the cow didn't have those microbes in its stomach, it would not be able to eat grass and get energy from that. So there's a specific part of the stomach where this happens, which is called the rumen. So a ruminant animal technically is an animal that has a rumen, which is a part of their stomach where the microbes live that digest grass for them. So when you have this cow eating grass, it's going to swallow that grass down into its stomach, and then it has a really complicated stomach with like 4 different chambers in it. So the food is going to circulate in here, and it's going to go, it's going to get, this large chamber is actually the rumen, where it's going to get digested. All the cellulose that's in that grass is going to get digested there, and then the whole thing is going to get upchucked and then swallowed again, so it could go down a different tube and enter the next chamber of the stomach, where those nutrients can actually be further broken down by the cow's digestive enzymes, and from there into the intestines. Right, but the rumen and the microbes in there is critically important for that process. So a similar thing happens in your own gut, where microbes are helping you digest certain foods, but the difference is that you don't absolutely rely on that for your ability to get nutrients that you need from your diet. The cow does. But it does affect you in different ways. So people who are lactose intolerant actually are not able, they don't have the enzyme to digest lactose, but there are microbes in their gut that can digest like lactose. So those microbes are going to start eating the lactose, and they have kind of a population explosion. And then since there's so many of them now, they can cause kind of intestinal symptoms of like nausea and diarrhea. So some of these organisms that are in your microbiome are like healthy for you when your microbiome is balanced and they have like a moderate population size. If their population explodes, then that can cause problems. That's an imbalanced microbiome. So your microbes can be like responsible for lactose intolerant symptoms, but theoretically, if we supplemented your microbiome with different microbes, then maybe you could actually fix lactose intolerance and hold the population of those kind of bad lactose-eating microbes in check. We don't understand the microbiome well enough yet to really do that, but one day we probably theoretically may be able to. Also, if you've ever had like sugar-free candy that has a lot of sugar alcohols in it, a sugar alcohol tastes sweet on your tongue, but you can't absorb it in your gut, so it doesn't go into your blood and basically just passes through. Except microbes in your gut can eat sugar alcohols. So if you eat too much sugar-free candy, then you can have similar symptoms as with lactose intolerance. Like bloating, nausea, diarrhea, that would be caused by kind of a little population explosion in the microbes that are able to eat those sugar alcohols. So you have to eat it in moderation. Microbes are also important for our food supply because microbes like to eat the same food that you like to eat a lot of the time, and they can make your food go bad. So that's, not good. We have a lot of ways to kind of prevent that from happening. So a lot of how we handle food is related to kind of preventing microbes from eating it or making them eat it slower. Right, so refrigerating your food, that is so that microbes will not be able to eat it. You're basically putting the food at a cold temperature and figuring that most microbes who would like to eat your food cannot live happily at that cold temperature, and so they're not going to eat the food, and that's generally correct. Curing or salting food also discourages microbial growth. Anytime you're drying, smoking, canning, pasteurizing, all of these things are ways to prevent microbes from eating your food. But you also have microbes that are helpful for producing food. Fermented foods are all foods that have been produced using microbes in one way or another. So there's a lot of foods like that actually. You have pickles, kimchi, cheese, yogurt, soy sauce, a bunch of sauces actually are all fermented foods, and you can't produce those things without microbes. Fermented foods actually, that's another way to prevent food spoilage. You're basically having like good microbes, basically microbes that can't hurt you, eat your food and make it tasty for you, and then bad microbes cannot eat it. It's already being eaten. So like kimchi, for instance, that was originally developed as a way to preserve cabbage through the winter time. You also have a lot of foods that are probiotic these days. Any fermented food actually could be considered probiotic, but you also have, well, I guess a lot of supplements these days that are probiotic, or like kefir is probiotic, or kombucha. So these are foods that are kind of fortified with beneficial microbes, and by eating them you can kind of replenish the microbes in your microbiome and ideally help balance your microbiome and make it stronger and more healthy. I'm sure it's possible to overdo it, but in general, having fermented foods in your diet is a healthy thing because it benefits the microbiome. Microbes are really important for industry too, both in bad ways and in good ways. So microbes can be harmful for industry because they're very diverse. They can eat a lot of things that you would not expect to be food. So if you have any type of like fluid that's traveling through pipes or it's stored in tanks, a lot of the time there's some type of microbe that will be able to eat that fluid or live inside of it. Right? So for instance, oil, you have microbes that eat oil. So you can get these microbes kind of contaminating your tanks or clogging up your pipes. You have to be careful about that and clean your stuff out, or find some way of preventing microbes from growing on your stuff. That's also really important in medicine or medical industries for implants or devices that you're going to put into someone's body. You have to make sure that you don't have microbes living on those things, because a lot of the time microbes will be able to live on them if you give them a chance. But on the flip side, microbes can also be used in good ways. So sometimes microbes will make stuff. If there's some chemical process that you need to conduct to make the chemical that you need for your industry, sometimes there's a way that, well, sometimes there's like a microbe that can produce that chemical for you that would be cheaper than producing it in other ways. Right, so you just get your batch of bacteria going. You give them the starting materials that they're going to use, and then you just sit back and they start making your product for you, and you just have to like purify it at the end. A bunch of things are made that way: vitamins, antibiotics, enzymes, many things that are complicated to synthesize. You can make easily and cheaply using microbes as basically little mini factories to make that thing. And microbes can also be used to clean up spills and waste products, since there are microbes that can eat basically any old thing that you can imagine. So if you have an oil spill, then you could introduce some microbes that can eat the oil and that can help clean up the spill. Bioremediation is the fancy name for using microbes or other organisms, technically just some type of biological thing, to clean up pollutants or spilled wastes. They're frequently used in water treatment plants, including here in El Paso. Actually, there's different points in the process of treating our water where microbes are used to remove pollutants and things from the water. So it's very beneficial when used correctly. So more detail on our wastewater. We actually have a very strong need to reclaim used water and treat it so that it's like safe for drinking once again, because of course we live in a desert and there's quite a lot of us here. We actually have a world-class water treatment system. So people will actually come from all over the world to tour our wastewater treatment facilities because they're really state of the art. And actually, the city has a strong investment in making sure that we have really strong water treatment facilities because of Fort Bliss. The Army doesn't want to have Fort Bliss here if there's not a secure source of water for everybody at Fort Bliss. So in order for the Army to sign a contract that says Fort Bliss is going to be here for the next X amount of years, El Paso has to prove to them that there will be water for that time. All right, so our economy and our lives, of course, are basically dependent on the quality of this water treatment system that we have. And of course, as time passes and climate change becomes increasingly severe, the area just gets drier, and so the need for a really efficient wastewater reclamation system becomes more and more pressing. So hopefully we're going to keep on top of that. This diagram here is basically an overview of our one of our water treatment plants, which is the Fred Hervey water reclamation plant. As you can see, it's very complicated. A lot of different processes are being done here to clean the water in various ways and remove different pollutants or contaminants from the water. But there's actually multiple places where you have microbes being used in this process. Kind of early on here, you have anaerobic microbes. Those would be microbes that do not use oxygen and a lot of the time can't survive oxygen. Those are being used to digest different contaminants in the water. And then again, in this whole big black part, there's different places in here where you're using microbes to digest different pollutants in the water. A lot of the pollutants that microbes would be digesting would be organic molecules. So those are they frequently are larger compounds that it could be hard to remove. They have a lot of sort of chemical diversity, lots of different chemical properties in the different organic compounds that can make them hard to remove, but microbes can chew them up. That would include also things like drugs, like you inevitably have some amount of prescription medication that's flushed into the water supply, and it can be pretty important to remove that so people aren't taking drugs that they're not trying to take. And microbes would be used to digest those things. And microbes are extremely important in science. They grow very quickly. You can have a lot of them without devoting a lot of resources compared to the amount of resources that it takes to house like larger animals. So it's easy to use microbes to do experiments. It's cheap, it's fast, it's easy. So microbes are used all over the place in biology in different experiments. Most of what we know about molecular biology is actually coming from experiments in microbes. So all the stuff we know about the different organelles in the cell, the different molecules in the cell, how all of that works together to run the cell, that's coming from microbes. Most of the stuff we know about genetics, that's come from experiments in microbes. Metabolism, that's coming from experiments in microbes. A whole bunch of stuff. So we're going to talk about growing microbes in the lab here in this class and also in our lab. So I'll go over some terms that are related to growing microbes in the lab for science or for industry purposes. So first we have the word culture. A culture is just a population of microbes that you are growing in the lab. So if you culture microbes, that's a verb. That is the act of growing them in the lab. If you have a culture of microbes, that's a noun. It's the population of microbes that you have grown in the lab. Then you have medium. A medium is just, it's food for the microbes, and it's like a place for them to grow on. So you can have different types of mediums or different types of media. In this picture here, you have, this is like a petri dish, and you have a solid medium in there. You can't really see it because it's transparent, but it's like a gel filling up this dish. And so that gel is giving the microbes like a surface to grow on, and it's also full of food for them. So it's giving them their food and all the different nutrients that they need to live. Here in this tube, you have a liquid medium, and it's full of bacteria. So like it's cloudy because you have a bunch of microbes, probably bacteria, growing in there. And so again, it's a place for them to live, and it has all the food and all the nutrients that they need to live. You can have a colony of microbes. If you have basically a cluster of microbes growing together that is big enough for you to see it, that's a colony. So on this plate here, all these little dots are colonies. Each dot has like millions of different microbial cells living in it. And it just looks like a little circle that you can see. If you were to take one of these colonies and zoom in, it might look like that. And if you zoomed in even further with a really fancy microscope, you might see something like this. These are all different cells. Like there's a cell, there's a cell, there's a cell. It's like millions and millions of little cells all jammed together to make this little colony. In these other parts of the plate, you're not seeing the circles because they're basically so close together that it's not looking like circles. It's looking like streaks or just a smear. Then finally, aseptic technique. Aseptic technique is kind of the way of doing things in the lab to prevent contamination with microbes. So basically making sure that the microbes you have growing on your plate don't end up in other parts of the lab or on your lab equipment or on your hands. and also making sure that little microbes that are kind of floating in the air don't end up growing on that plate. So aseptic technique is really important for basically preventing microbes from traveling freely around the lab or in and out of the lab, and for controlling where they are, where they're growing, and for keeping all your things clean and uncontaminated. Part of aseptic technique is washing your hands. So we're all kind of practicing aseptic technique right now in a way, with all our frequent hand washing and all of our washing our doorknobs and sink faucets and everything that gets commonly touched. That's all aseptic technique. When you are careful about what you're touching because you know that you just got your hands maybe contaminated, and so you're going to use hand sanitizer before you grab your food, that's aseptic technique too. So all of us are practicing it now, and we'll get a chance to practice it some in lab as well. So now we're going to talk about microscopy. Microscopy is really important for the study of microbiology because, of course, you can't see microbes unless you have a microscope. So the development of microbiology has really been dependent on microscopy. Before you had microscopes, you couldn't have microbiology. There was no way for us to know that microbes even existed. And as microscopes have gotten better through the years, you've been able to see more and more of these microbes, and we've been able to learn more and more about them. So microscopes have a couple of different, functional properties. First, they have magnification, which is actually making the image in the microscope bigger. So how much magnification the scope has is how many times bigger the image in the scope appears versus how big it really is. Then you have resolution also. Resolution is like the resolution on your TV screen. It's how clear things look. So if your microscope has a big magnification, but it has a small resolution or a bad resolution, then you're going to see a large image, but it's going to look grainy. It's not going to look nice. You have to have a good resolution to really see details in the image. Technically, resolution is how far apart things need to be before you can see that they are two different things. So if you have two dots, if you look at them close up, you can see that they're two different dots. But if you put them on the other side of the room from you and go across to the other wall, maybe you don't see them as two dots anymore. You just see them as one dot. There's not enough resolution in your eye to still see at that distance that it's two different dots. You have two basic types of microscopes. First, you have the light microscope that was developed first, and then more recently, we invented the electron microscope. The light microscope is using light to magnify the image, and the electron microscope is using electrons to magnify the image. And that's the main difference. And electrons are abbreviated as E-. The electron microscope is much more complicated. It's newer. And it has a much better magnification and much better resolution. So it lets you see things that are really, really, really tiny, like ridiculously small. The reason that works like that is because electrons have a wavelength that's smaller than light waves, than the wavelength of light or photons. So more detail on a light microscope. These are the basic parts of a light microscope. You have the condenser. Let's see, on this image of a light microscope, the condenser is right here. It's beneath the stage where you would put the specimen. The condenser is gathering light and focusing it on the specimen that's on the stage. Then you're going to have lenses. Directly above the stage, you have your objective lenses. And then you also have your ocular lenses that you would actually look through. So those lenses are going to be magnifying the image. Both of them magnify, actually. The oculars and the objectives both magnify. And there's this number, numerical aperture, which is kind of describing how much light the lens can gather, or how big a space the lens can gather light from. The more space that the lens can gather light from, the bigger the image that it can make. On microscopes that you use for microbiology, you have these objective lenses. A lot of the time, one of them, or always actually for microbiology, one of these objective lenses will be what's called an oil immersion lens. It'll usually be the 100x magnification lens as an oil immersion lens. What that means is you need to put a little drop of oil on the lens or on the specimen so that the light is passing through oil on its way into the lens. And that is necessary to get a clear image with the oil immersion lens. Basically, this oil has a different refractive index than air, and so it bends the light differently from air. So if you have a light beam passing through the condenser here to the stage, passes through the specimen, as soon as it's going through the air, it's going to start bending, and it might just bend completely off target so that it doesn't go into the lens at all. If you have that little drop of oil there, though, it's going to bend differently. It's going to bend more like it bends through glass. So you will have your light wave or your light beam traveling up through the condenser, through the specimen, and the oil kind of holds its path steady, and then it ends up in the lens. And if it's in the lens, that means that ultimately you can see it. So whenever you're actually using a microscope with the oil immersion lens, it's really important to remember to clean the oil off that lens when you're done. Otherwise, it's going to get onto the other lenses potentially, and they are not designed to work with oil, so they're going to have a blurry image if they have oil on them, and it can be really hard to get that oil back out again. Something to bear in mind if you're ever with a physical microbiology scope. In general, the maximum magnification for a light microscope is 1000x. That would be with the 100x objective, and the ocular lenses will usually magnify it an additional 10 times. And the maximum resolution will be 0.2 micrometers, which is real small. 1 micrometer is 1,000th of a millimeter, so 0.2 micrometers is like 2 10,000ths of a millimeter, so it's real small. You also need in light microscopy to have something that introduces contrast between the specimen you're trying to look at and the background of the stage or the slide, because these microbes in general are like transparent. So if you don't have something to give them some color, some contrast, it can be real hard to see what you're trying to look at. Staining is used to give contrast to these images. So the process of staining is just applying dyes to the specimen to increase the contrast. Stains would usually, a lot of the time, kill the specimen. So depending on what purpose you're looking at it for, sometimes you can't stain it because you need it to be alive. But we usually use stains. So the stain or the dye that you're going to use for the stain needs to have two things. It needs to have a color, and it needs to be able to stick to your specimen. And different dyes will do that in different ways. A lot of the time the dye has a charge, an electrical charge on it, like a plus or a minus, and so it sticks to anything that's on that specimen that has an opposite charge. You also have special dyes that are fluorescent, which means that when you just look at them, they don't have a color, but when you shine light on them, that's the right wavelength, they will then start to glow. And then you kind of have to turn like the lights off in the room so you can see the glow. And so that's pretty cool. So this image here is actually using fluorescent staining. And all these are stains, of course. Then you have differential stains. A lot of stains are differential stains. Any staining method that uses 2 or more dyes is differential. So this fluorescent stain, that's a differential stain because we've got 3 different colors here, 3 different dyes. Each of those dyes is labeling something different inside the specimen. So you have one, that's the blue one, that's showing the nucleus of each of these cells. Then you have another that's red, that's showing ribosomes of the cells. And then you have another in green that's showing the cytoskeleton, which gives you kind of the basic shape of the cell. So differential stains can let you see more information and more detail about the specimen. This is also a differential stain over here. Here you've got these blue balls. Those are all cells. This is actually a slice of tissue. So these would be like actual human tissue body cells. And then in the red here, you've got all these tiny little rods. Those are actually a bacteria that is infecting the person's cells. So by using these two different dyes in the staining process, you can see the patient cells and you can see the pathogen that's infecting them. There's one type of stain that is super common in microbiology, which is the Gram stain. The Gram stain is a type of differential stain because it uses two dyes, and it's basically done all the time. It was invented in 1884. It was a pretty early stain, invented by Hans Christian Gram, which is why it's called the Gram stain. So the Gram stain lets you differentiate bacteria based on their cell wall. Some bacteria will stain purple with the Gram stain, and we call that Gram positive or Gram plus. Some bacteria will stain pink or red, and we call that Gram minus or Gram negative bacteria. The main difference between Gram positive and Gram negative bacteria is the cell wall that they have. So we'll go into more information on the cell walls in the next chapter, in chapter 2. But the most common types of cell walls that a bacteria can have are either the gram-positive wall, which has these it's real thick. So actually at the bottom here is just the cell membrane, and then this part is the cell wall. All this kind of reddish, pale reddish stuff is a molecule called peptidoglycan. You just have a bunch of sheets of it real thick in the gram-positive cell wall. And this would stain purple with the Gram stain. The gram-negative cell wall looks different. So at the bottom you have the cell membrane, and then on top of it you have the cell wall. So the cell wall has a little bit of the peptidoglycans, but it's pretty thin. Then above that it has a second membrane, an outer membrane. And this will all stain pink or red in the Gram stain and be called Gram minus. So when you do a Gram stain on bacteria and you see that some of them are purple and some of them are pink or red, then that's already telling you something about their cell walls, which is very useful because it's a simple stain, it's quick to do it, and it lets you see real easy which ones would have a Gram-positive cell wall or a Gram-negative cell wall. And that ends up being very useful in medicine because antibiotics will a lot of the time only work against gram-positive or gram-negative bacteria. So you wouldn't want somebody who has a gram-negative infection to get treated with antibiotics that are only good for gram-positive bacteria. That's not going to help them get better. So you want to do a sample, you want to take a sample of their infected tissues, culture those bacteria, get a sample of them, and stain them to see if they're gram plus or gram minus. That helps you know what antibiotic to give. So we'll go real quick through the basic principles of Gram staining. These are similar actually for a lot of stains, starting with fixation. So basically a huge amount of stains start with fixation, where you're basically, you have your slide, it has a sample of bacteria on it, and you're going to heat it to fix the bacteria onto the slide so they don't leave or get washed off later on. Then you start the staining process. So first you're going to stain by submerging that slide in crystal violet dye. That dye is going to stain all the cell walls and it's purple. Then you apply iodine, which actually it kind of grabs the crystal violet and you get these complexes of crystal violet and iodine that are kind of embedded in the cell wall and they kind of get stuck in there. So it makes the cell wall, the crystal violet makes the cell wall purple, but the iodine sticks the purple on there so it can't come off. But of course the gram-negative cell wall doesn't have very many layers for the iodine and crystal violet to get trapped in, so you're not getting very much purple trapped there. But in the gram-positive cell wall, there's a lot of layers and you get a lot of crystal violet trapped in that cell wall, making it very purple. The next step is alcohol. Alcohol is actually going to remove the crystal violet, so it takes it off, unless it's trapped in that cell wall with the iodine complex. So your gram-negative bacteria, they don't have a lot of layers in their cell wall. They're going to have most of their crystal violet removed by the alcohol, whereas the gram-positive will have most of their crystal violet stay behind because it's trapped in those layers. Then finally you stain with safranine, which is kind of pink-red. Whether it's pink or red depends on kind of how much you put and how long you leave it for. It can also depend on other vagaries of the staining process, but it's going to stain pink. So anything that had the crystal violet washed away, right, if your gram negatives would have the crystal violet washed away, and then you add the safranine, and then they end up looking pink or red. Your purple ones, the gram positives, never had that purple washed away. So at the end, when you stain with safranine, they do have safranine on them, but they still look purple because it's just a stronger color and there's more crystal violet than safranine on those cells. So we'll kind of briefly go through the different types of light microscopy. First you have bright field, which is sort of the regular type of light microscopy, where light just goes through the specimen and is collected by the lenses. The problem with bright field is that a lot of the time you don't have good contrast because your specimen is transparent. So you need to do different things to improve the contrast to get a good image. Commonly that would mean staining with bright field, but you could also use a different type of light microscopy to get contrast without staining. So you can use dark field, for instance, where you're only collecting light that has scattered off of the specimen. That's the light that would actually be creating the image of the specimen. Other light that is not scattered would be coming from the background, and that gets blocked with dark field. So what you get is a light specimen and a dark background as a result in dark field. You also have phase contrast light microscopy, where the light that is scattering off of the specimen that makes the image is just handled differently from the background light that is not scattered. So there's different types of phase contrast microscopy actually, where the background light would be handled in different ways, but in general it's kind of the wavelength of that background light is going to be shifted and then blocked out or else enhanced, so that you are going to end up with the image looking different from the background. In general, you're going to get a light background and a dark image with phase contrast microscopy. We won't worry about the details of it so much. You also have differential interference contrast, or DIC, light microscopy, where you have polarized light. Polarized light is light waves that are traveling in a specific orientation, and you're manipulating the polarized light and putting it back together at the end, like separating light into different polarizations and then putting it back together at the end, and it gives you a 3D illusion for the object that increases the contrast. You also have fluorescence microscopy, where you're using very little light really, so the background appears dark, and you're using fluorescent stains on your specimen. So you would be shining a specific wavelength onto the stain, causing it to glow, and then you're collecting light that's only at that glowing wavelength. So you see a dark background with a colored image. Then you also have confocal scanning laser microscopy, which is similar to fluorescent microscopy, so it also uses a fluorescent stain, but you're using a laser instead of just a regular light bulb, and you're using a little pinhole to block out some of the light that's coming from different parts of the specimen, so you're only looking at one layer of the specimen. So it gives you a very high resolution image. You can also recreate a 3D image using confocal scanning laser microscopy. So you can take an image at the bottom of the specimen, then a little bit up from the bottom, and a little bit up from that, and a little bit up from that, and then you can combine all those images together to make a 3D model. So these are examples of images that are taken with those different techniques. So first we have bright field down here. So that's this is bright field without staining. As you can see, the cells are basically the same color as the background. So you can see them, but it's kind of hard to see details in those cells. And then here you have the same image but using dark field, and you can see there's better contrast between the cells and the background, and you can also see the different details within the cells better. And here's with the phase contrast microscopy. And so you can see there's even more contrast compared to the dark field and to the bright field, and the cells are like glowing basically. Then here you have the DIC microscopy with basically a small 3D effect given to these cells, so which makes them stick out really clearly. In the corner you have fluorescent microscopy, so these cells have been labeled with a fluorescent stain which makes them show up brightly. But as you can see, there's still, this image is still kind of blurry. If you use confocal scanning laser microscopy, then you get a much clearer image with the fluorescent stain. Next we have electron microscopy. There's two types of electron microscopy, transmission and scanning. Transmission electron microscopy is more like bright field light microscopy, where you have your beam in electrons, your beam of electrons in this case, passing through the specimen and being collected. So this allows you to see the structures that are inside of your specimen. So it can actually show you like organelles and different structures inside of cells with extremely large magnification and very small, very small resolution, which gives you a really clear image of even very tiny structures. One of the main limitations with transmission electron microscopy is that you need a section of that specimen that is extraordinarily thin. So they have like specialized machines that you use to cut up a specimen into thin slices so you can get light going through it for microscopy. And it's basically like a super fancy like sandwich slicer or a deli meat slicer, like if you've seen at a deli the machines they use to slice meat into sandwich slices. That's similar. It's like that basic type of machine. The one that they use to cut sections for electron microscopy, these sections are so thin that that machine has a microscope on it actually, so that you can see what you're doing because it's so small. Then you have scanning electron microscopy, where the beam of electrons that you shine onto the specimen, you're shining it down from on top actually, and then the beam is going to bounce off the surface of the specimen and instead of going through it. So this is showing you the terrain sort of or the topography of the specimen surface, or the basic outside shape of the specimen. Also with extremely high magnification and small resolution. In electron microscopy, you always get images that are in black and white. There's no color. A lot of the time you'll see electron microscopy images that have color. That's all artificially added color. So they've added the color in later. And the quality of electron microscopes are constantly improving as technology gets better. Okay, so now we're going to go over some of the history of microbiology. And see how it parallels kind of the development of science and important questions, scientific questions. So before we get into this, I'll just make a quick note. The science history is messy. So the version of science history stories that's presented in textbooks is always like very simplified. Right, so in the textbook you'll read that, like, you know, such and such a person invented this thing or came up with this idea. Actually, in real life, multiple scientists would have worked on any given thing, especially when it comes to theories. Each theory is the result of many scientists all working and getting new information, tweaking the theory, and advancing it. But at the end, it'll be like one or maybe two people who gets the credit for that theory, even though very many people have worked on it. So in general, someone will propose an early version of the theory, but there's no evidence yet, and it's kind of seen as like a crazy idea. Then as time passes, others will take that idea and develop it and tweak it, changing it. You'll have new evidence emerging, and most importantly, new techniques emerging as technology improves. And those new technologies and new techniques let you get more evidence that would start to support the theory. Then you can continue tweaking the theory with reference to that new evidence that's coming out. And then at some point, somebody is going to make either a significant change to the theory or get really conclusive, like clinching evidence that supports the theory. And that's the person who gets credit for the theory. And then even after that point, usually other people will continue to develop and tweak the theory. So theories are always kind of like living things in a way, where they're always being developed. Sometimes you'll have different people working in different parts of the world that propose like a really similar idea at the same time, independently of each other. In that case, we're only going to remember one of those people. So for instance, you had Charles Darwin propose the theory of evolution. At about the same time, another guy, Alfred Russel Wallace, working on the other side of the world, also proposed evolution. But Charles Darwin got credit for it because Charles Darwin had a book full of evidence. Alfred Russel Wallace only had the idea. Sometimes stuff that's not really related to a scientist's work will impact how they're remembered or how their work is remembered or how their contributions are remembered. So sometimes scientists would get into fights with other scientists that results in their reputation being damaged, and then they don't get credit for their work. Or if they later go on to work on something else that people think is like silly or looked down on. Or if you're not the right demographic. So historically, scientists that have had their work kind of acknowledged and remembered have been white men. And if you weren't in that demographic, it would have been harder for you to get recognition for your work. So just keep all of that in mind as we go through science history stuff right now. So microbes were discovered after the light microscope was invented. No one really knows exactly when it was invented, sometime between 1590 and 1620, and there's actually multiple people who claim to have invented it, and we don't really know who did. But somewhere in there, the light microscope was invented. And after that, people started using it to look at really small things and to draw those things, and eventually microbes were discovered. So this guy, Robert Hooke, is one of the earliest people to look at things in the microscope, draw them, and then publish that in a book. And he actually discovered cells, and he discovered reproductive structures on molds that's actually a type of microbe. So that was published in 1665. So he didn't really understand the significance of what he found. He just thought he found like some interesting looking stuff, but it was actually a living thing, a microbe. So here's Robert Hooke's microscope, and here's a drawing he made of the mold reproductive structures that he saw using his scope. Then microbes were like kind of properly discovered by this guy, Antony van Leeuwenhoek, who's generally credited with discovering microbes. He actually made his own microscope lenses, which were very advanced lenses for the time, and he could see things with better magnification resolution than anyone else could. So he actually discovered microbes. He understood that they were alive, and he drew them and published that. So he discovered all the types of microbes except for one, viruses, which are very, very, very small. You can't see them with a light microscope. You have to have an electron microscope. So he wasn't able to discover those. But every other type he was able to find. And these are all drawings he made of the different microbes that he discovered. He didn't call them microbes, he called them animalcules or beasties. And he did know that they were alive. And this is a drawing of the microscope that he used. So actually, he was kind of a jealous guy, jealous of his work. So he wouldn't tell anyone how to make the lenses, his special lenses. And he wouldn't even let someone look through his microscope, actually. Like, if you wanted to see what he was seeing, you just had to look at his drawing. He wouldn't let you look through the microscope. And when he died, he hadn't told anybody how to make the lenses. And so the whole field, basically, of microbiology was set back for about 100 years until someone else was able to make lenses that were the same quality as what he had been able to make. So he published his discovery of microbes in 1674. Right, and then he died the secret of the lenses went with him to his grave, and it's hard to study something when you can't see it, and it was impossible to get good quality images of these really small microbes without his special lenses. So the field kind of languished for another 100 years until lenses of the same quality were invented by someone else. At that time also, no one really thought that microbes meant anything or that they were important in any way. It was just seen as kind of like a curiosity, basically. So eventually when microbiology was revisited by scientists, these scientists were trying to answer two big questions about life. First, where do living things come from? And second, what causes disease? So first, where do living things come from? For many centuries, people thought that living things come spontaneously from non-living things. And this idea is called spontaneous generation, and it was the leading theory since the ancient Greeks, basically. So people actually thought that, you know, bugs would come from food that was left out. So if you had like meat that went bad and maggots were on it, people thought that the maggots came from the meat. And this is like a recipe for growing mice. So in this recipe, you take sweaty underwear and stuff it in a jar with wheat, and you wait 21 days, and then you get mice. So the idea would be that the wheat and the underwear spontaneously generated these mice. Of course, this is not true, but at the time they didn't know. So at long last, it took a long time to disprove this theory actually, but at long last it was microbiology that was used to disprove the theory. And the disproving of spontaneous generation is also really important in science history because it led people to formalize the scientific method for the first time. So people were kind of using it before, but they hadn't like written down the steps of it, which means that they weren't completely following it exactly. And so this is kind of a revolution for science. So one of the first experiments that was used to disprove spontaneous generation was by Francesco Redi in 1668. He did this experiment where he had some jars with meat in them. He left one just open, another one he sealed with some cork, and then the last one he put some gauze over it and sealed it that way. And he just basically waited to see what would happen to this meat. In the jar that was open, of course, you had maggots. Flies would come onto the meat, they laid their eggs, and you got maggots. He didn't understand that that it was like flies laying eggs, but he, understood that it was open to the air, and flies came in, and there was maggots. On the ones that were sealed, you did not get any maggots growing, and flies didn't, of course, enter those jars. But on the one that was covered with gauze, he noticed that you would have flies come and just sit on the surface of the gauze. And that eventually you got maggots on the gauze itself. So at that point he figured, it's the flies. It's the flies somehow leaving these maggots. It's not the meat itself. If it was the meat itself, you would have maggots on meat that was in sealed jars, and you don't. So that basically disproved spontaneous generation for things that you can see, but the theory still held true, or was thought to hold true, for microbes. For a variety of reasons. One being just that Leeuwenhoek, one of the things he had looked at was raindrops in his microscope, and you would see that at first the raindrop is clear, and as time passes it gets cloudier, and you can see little microbes inside of it, or little beasties inside of it, and so he thought that they came from the rain. And there was also another experiment by John Needham. He boiled beef broth, and he put it in a jar and sealed it with cork. And he saw that over time the broth became cloudy as microbes started growing in it. That was published in the 1740s. So that experiment seems to support spontaneous generation. It seems like the microbes came from the beef broth, but actually there were some issues with the methods he used for that experiment. So he didn't boil the beef broth for very long, and then he kind of let it sit open to the air while it cooled, and then he put it in jars and corked them. So there was plenty of time for microbes to be reintroduced to the beef broth from the air before he sealed it. But he didn't know that at the time. So next you had this guy, Lazzaro Spallanzani, which he kind of repeated John Needham's experiment, but he tweaked it to make it more precise. And he published that in 1799, so like about 50 years later, actually. So what he did is boil the beef broth. Right, so here he has beef broth boiling. He boiled it for a longer amount of time, and right after it was done boiling, he sealed the flasks. Of course, he had some flasks that he did not seal, he left them open, but some flasks he did seal. And he sealed them right away when it was not cool yet, and he didn't seal them with cork, but he actually melted the glass in the neck of the flask closed. So that's super sealed. And he saw that in these flasks that were sealed, he didn't get cloudy broth. In the flask that was open, the broth did become cloudy, which of course suggests that you have microbes in the air falling into the flask and then breeding there in the broth. So that's a pretty good experiment, but other scientists did not want to accept it at the time. They said that there's stuff wrong with his methods. So they said that he boiled it for so long that it was too long. That he actually kind of boiled it so long that he destroyed the ability of the beef broth to support life in some way. And they said that because he sealed the flasks by like sealing the actual glass, that there wasn't enough air in the flasks left for microbes or for life to live in them. So people didn't really want to accept his results. Which meant that you needed yet one more experiment to finally disprove spontaneous generation. And that one was by Louis Pasteur in 1861. So again, actually a bit more, like 60 years later this time. So Louis Pasteur is famous for a lot of things, but one of the things that he gets credit for is disproving spontaneous generation, because out of the many experiments that were done to disprove spontaneous generation, his is the one that was accepted by the scientific community. So he did a similar experiment to Spallanzani, but again, he tweaked it up. So he boiled his beef broth, he put it into the flask, and then instead of sealing the flask by melting the neck closed, he actually heated the glass and pooled it into an S shape. So after he got the broth into the flask, he heated the neck up and pooled it into an S. And he figured that it's sealed that way. So basically, if you have something in the air, it can fall into the flask. Here you have stuff in the air falling into the flask, but it's only going to go into the neck because it's going to get stuck in this little U. It's not going to make it all the way into the body of the flask that has the broth in it. But air can still get through. So people who are saying there's not enough air, those people were quieted by this because air still has access to this flask, but stuff that's in the air does not have access. So it is effectively sealed. And he left these flasks for 18 months, like under his stairs. And at the end, it was still like clear beef broth without microbes growing in it. What he did at the end actually is tilt these flasks like so that the broth kind of ran down into the neck, and then he put it back upright. So when the broth runs down here, it's going to pick up some of these microbes that have fallen into the U of the neck, and it brings them back when he puts it back upright. It brings it back into the main body of the flask, and at that point he got cloudy broth, which conclusively demonstrated to the satisfaction of everyone at the time that actually these microbes are coming from little particles in the air, which are themselves microbes, and it's not coming from the beef broth. Pasteur also had some other important contributions. He studied fermentation. So there were a number of alcohol makers who were having trouble making alcohol. They were finding that sometimes they try to make a batch of alcohol and it's just like acid, it's just like vinegar basically, and it's not useful for them. So they wanted to know, like, why is this happening and how can we stop it? And Pasteur was able to help them out and figure it out. So he did a series of experiments where he took these flasks that had grape juice in them, and in some of the flasks he introduced bacteria, in some of the flasks he introduced yeast, and in some of the flasks he didn't put anything. And he just saw what happened, and he found out that when you put bacteria in the flask of grape juice and wait, and seal the flask and wait, over time you're going to get vinegar. When you put yeast into the flask and seal it and wait, over time you get alcohol. So based on this, he was able to figure out that when you're trying to make alcohol and instead of getting alcohol you get vinegar, that's because you have bacteria in your grape juice. Your stuff is contaminated with bacteria. You need to just have yeast, only that. And so following this insight, he was able to tell these alcohol makers that they need to clean their stuff real good, basically. So if you had a batch of alcohol that was supposed to be alcohol that turned into vinegar instead of just reusing that flask, you have to clean it out. They didn't know you have to do that, so they were just getting vinegar every time. Once they started cleaning their materials, then they had much better success with getting alcohol and not vinegar. And he also invented the process of pasteurization, which is used to keep things sterile, basically, without destroying them. So, of course, people knew that if you boil something, you're going to get rid of the microbes and it's not going to go sour. But a lot of the time, if you boil something, you kind of ruin it, depending on what it is. So milk is a good example. You can't boil milk. It won't be good anymore. And so this process of pasteurization is designed to just heat that substance like milk just enough to kill almost all the microbes, but not enough so that it boils and the milk is like destroyed. And that's the same process that we use today to sterilize milk and a lot of other things. You can see on a lot of packages at the grocery store that they say they've been pasteurized. So we'll go over the scientific method really quick because it was developed formally during this debate over spontaneous generation. So in the scientific method, you start with observations about the natural world. Those observations spark your curiosity and you end up with a question that you want to have answered about what's going on. Then you make a hypothesis, which is a possible answer to that question. The hypothesis needs to be stated in a specific way. So it needs to be testable and falsifiable. It needs to make a prediction that you can test. So if you have a hypothesis that's impossible to test, like if your hypothesis is that the tooth fairy lives on the dark side of the moon, there's not a way to really test that, so that's not a good hypothesis. It has to be something that you can test and find out that it's right or it's wrong. So once you have your hypothesis, then you design an experiment to test the hypothesis. So you're going to figure out what prediction your hypothesis makes, and then do an experiment where you see if that prediction comes true. And anytime you're running an experiment, you have to be careful to include control groups. So whenever these guys working on spontaneous generation did an experiment and they boiled some broth and put it in a flask and sealed it, they would also have some broth that they boiled and put it in a flask and left it open. That's the control. So you can see what happens without your sort of experimental intervention of sealing the flask or whatever else it is that you're doing. Right, so you do your experiment and then you observe what happens in your experiment. You collect data from your experiment and then you're going to analyze that data. When you analyze the data, you find out either they do support your hypothesis or they do not support your hypothesis. If they support your hypothesis, then you get to accept your hypothesis, but you're still not done because you need to repeat. In order for a hypothesis to be truly accepted as true, then it needs to not be based on just one experiment. You have to have a lot of people doing the same experiment, and even better, different experiments, using even just different techniques that all have the same results, and that is really strong evidence that your hypothesis is correct. And then eventually, if there's enough evidence, it could become maybe a theory. If your data do not support the hypothesis, you're going to reject that hypothesis. Then you're going to modify it, basically make a new hypothesis, and test that modified hypothesis. So the next question that microbiology was used to answer is, what causes disease? So again, for many centuries since the Greeks, there were these ideas about where disease come from. The two main ones were humorism and miasmatism. Humorism is the idea that health is about balanced humors, and the humors are just these four body fluids. that people thought need to be in balance in order for you to be healthy. They were blood, yellow bile, black bile, and phlegm, which is spit. So if you had the right balance, you were healthy. If one of them was too much or too little, then you became unhealthy and you got a disease. And people thought that the humors were also related to your personality and even what your face was shaped like. So here, these are like different head shapes that supposedly were associated with different combinations of the 4 humors. The other leading idea was miasmatism, which is the idea that diseases are caused by bad smells, smells that's coming from like rotting things or sewage, basically. So, of course, we know that sewage and rotting things have microbes in them that can cause disease and that can be transmitted through the air, but in miasmatism, that wasn't known. They just thought that literally it's that bad smell. So, if you cannot smell it, then you cannot get sick. That's why these kind of medieval age doctors wore this crazy, like, creepy bird mask looking thing. That's actually stuffed with, like, potpourri, basically, in the beak. It's all stuffed with potpourri. And so the purpose of it is to keep the bad smell out. And they thought if you don't smell it, it's all good. So over time, as people began to study microbiology, eventually we realized that diseases are caused by microbes, by certain microbes, which is germ theory, basically. The disease is caused and transmitted by these little particles, which are microbes. Germ theory was first proposed in 1546 by Girolamo Fracastoro. He just thought they were little particles. He didn't know that germs were alive, or he didn't think that germs were alive. And he didn't have proof for this theory either. Like, he had no evidence. It was just a good idea that he had. But people at the time didn't think it was a good idea. They thought it was a crazy idea. They believed in miasmatism more so. So the theory kind of languished until Pasteur again took it up in 1857. Well, actually, to be honest, you had a lot of other scientists working on it as well, but Pasteur is again the person who's frequently given credit for germ theory. And he actually proposed in 1857 that these little particles aren't just inanimate things, but they're microbes and they're alive and they cause disease. Today we conceive it a little differently. So today in germ theory, today we understand that germ theory only applies to diseases that are infectious, right? So there's other diseases like heart disease or, diabetes. Those are not infectious diseases. They aren't caused by microbes. They're caused by other things. So germ theory is only for infectious diseases that can be transmitted from one person to another person. And here you have like an early cartoon making fun of germ theory. The caption for the cartoon is "the latest contributions in the germ theory," and you have like an early scientist in his lab looking at a small man in a like magnifying glass, and basically this is trying to ridicule the idea that germs would be living things that cause disease by saying that we're saying there's like tiny people that make disease. Like, silly. So you also had another guy make really important contributions to germ theory, who is Robert Koch. And he's the one who finally proved that germ theory is correct to the satisfaction of other scientists. So sometimes you see Louis Pasteur credited with germ theory, and if not, it would be Robert Koch who's credited with germ theory. He actually discovered not only that germs cause disease, but specific microbes that cause specific diseases. So he was able to identify the specific bacteria that causes anthrax and tuberculosis and cholera using his microscope in the late 1800s. He invented a lot of techniques for culturing microbes in the lab, which hadn't really been done previous to his work. So he tried using different things as media to culture microbes, but finally he figured out how to use agar, which is a substance derived from algae, actually. So agar is a great growth medium because at room temperature it's solid, it's like a gel, and it has a lot of nutrients that bacteria need, so they're happy to grow on it. And he was actually able to have these little plates, like plates filled with agar that grow bacteria, and so he saw colonies. And he was looking at colonies and realizing that different colonies kind of look different from each other, and he was able to use the way that colonies looked to identify different species of bacteria. All right, so here's one type of colony versus another type of colony. Like, these look very different. They're made by different species. So he could identify, like, if you see a colony that looks like this, you know what species it is. So he was able to make these series of postulates, 4 postulates that are called Koch's postulates, which are, it's basically 4 things that you have to do in order to conclude that a given disease is caused by a given microbe. So you have to satisfy all 4 postulates before you can say that this disease is caused by this microbe. This is all disease etiology. Disease etiology is the cause of diseases. So anytime you're looking at what causes a disease, you're investigating the etiology of that disease. And his 4 postulates are, first, the suspected microbe has to be found in all sick hosts and not in healthy hosts. So you have to find it in all the sick people, and you can't find it in healthy people if you're going to say that it causes the disease. Next, you have to be able to isolate that microbe and grow it in the lab. Basically, you have to be able to culture it. Then you have to take your cultured microbe and give it to someone, not a person, an animal, give it to an animal who is healthy, and then you have to see that animal get sick with that specific disease. And then finally, you have to be able to take that new sick animal, take a sample of the microbe from it, culture it again, and you have to look at it and see that it's the same. When it makes colonies, it looks the same as the colonies that you first got when you cultured that microbe, so you make sure it's the same thing. If you can satisfy all 4 of those conditions, then you can say with a very high degree of confidence that this disease is caused by this microbe that I thought is the suspect. So these postulates have been used by all types of scientists to identify the microbes responsible for different diseases, and they are still used today, actually. The only exception would be, well, I guess at this time when Koch developed these, they were used to discover the responsible agents for many different diseases, where those agents were bacteria. But he wasn't able to discover the agents that cause viral diseases this way because they just didn't have the technology at that time. So here's a kind of a graphical representation of Koch's postulates. So first, you have to identify the suspected microbe in diseased animals, but not in healthy animals. So if this is some type of disease that affects mice, you should be able to look at healthy mice, take samples from them, and see no microbes. Then when you take the unhealthy or diseased ones, and you take a sample, you should see the microbe, these little rods. Then you should be able to get that microbe and grow it in the lab. Once you've grown your microbe, then you have little colonies of it. You can take some of those colonies and give it to another animal who is healthy, and then you see that animal get sick. Then you take a sample from that animal, find the microbes and get them, grow them in the lab again, and you have to see that the colonies from this new sample match the colonies from your first sample. Then you know with a high degree of confidence that that microbe caused that disease. So another important thing in the early history, well, early-ish history of microbiology was the development of aseptic technique, which developed alongside germ theory. And you had 3 people who made really important contributions to aseptic technique at different times in this period. So you had this guy, Ignaz Semmelweis, who was the first person to suggest that doctors should wash their hands. So in the hospital where he worked, you would have doctors who were doing autopsies of people who just died of disease, and then immediately from an autopsy, they would go and deliver a baby. and a lot of the time the mother would get sick and die, and no one really knew what was happening. But he had this idea like that you are contaminating your hands with this corpse, and then you're touching like the mother who's just, you know, given birth, and so you're introducing like a microbe into her that is then making her get sick and die. And what you should do to prevent that is wash your hands. And indeed, it worked. Even though it worked, a lot of people did not accept it when hand washing was first introduced. They still thought it was silly, and made fun of him for it, actually, before everybody accepted that you should wash your hands. You also had this guy, Joseph Lister, who introduced using antiseptics to sterilize stuff, to clean your tools and your bandages and everything that you're going to use on a patient. This antiseptic that he used was carbolic acid. So today we would use things like iodine or alcohol, or bleach or different stuff like that to clean stuff and make sure microbes are not living on it. Then finally you had a woman, Florence Nightingale, who was a nurse, who introduced a lot of hygiene practices, a lot of different ones. Just each one has a small impact, but then it all adds up. So one example of a practice that she introduced would be just storing clean clothes separately from dirty clothes. So, you know, when the patient like bleeds and has diarrhea all over like their bedding, you don't take that bedding and put it right next to the clean bedding. You put them separate. And you know, stuff like that makes a big difference in the hospital. And so for all three of these people, the changes that they introduced met with resistance. So people didn't want to wash their hands and use antiseptics and store stuff separate. They didn't like it, but you know, it worked. So eventually people had to admit it works and we should do it. The picture here is a plate where you actually have someone just put their hand on this plate. They literally just stuck their hand onto the agar there and then just saw what grew, and this is what grew. So you can see it's quite a lot of microbes that you have on your hand. Some of these would be from your microbiome and some maybe not. And you can almost see like the fingers here from the handprint and the thumbs right there. So your hands are very dirty. They're the dirtiest thing you have. Definitely wash them a lot. So that's basically the history of microbiology. In the modern era, sort of in the 1900s and up till now, microbes were used to make all types of discoveries in so many different fields related to biology, primarily like ecology, biochemistry, genetics, molecular biology, and they've been used in technology, the technology of genetic engineering. So we're actually going to get a little bit more into that genetic engineering technology later in the semester, but you can do a bunch with microbes. They're very, very useful. So one thing you can do is take genes for human proteins and put them into a microbe, and then that microbe will make that protein for you. So for instance, people who have hemophilia, that's a blood clotting disorder where they can't form blood clots properly, and so they have a tendency to bleed a lot if they get cut, and they can actually bleed out. How to fix that? We didn't know how to fix it until we were able to do genetic engineering. We were able to take genes for these clotting factor proteins that it turns out hemophiliacs are missing. You take the genes for those things, you put them into a microbe, and you make that microbe mass manufacture the clotting factor, and then you can just inject it back into the hemophiliac and they will be fine and their blood will clot normally. Same thing for insulin. For diabetes, like there wasn't a way to treat diabetes really before you had this technology where the human insulin gene can be put into a microbe and the microbe will mass manufacture the insulin protein and then you can give it to diabetics. So in that technology, it's kind of shown in this diagram here. You have your human cell and you're going to get a gene out of it that you need that protein for someone who's sick. And then you have a bacterial cell and you need a plasmid that's in that cell. You're going to cut that plasmid open, put the human gene in, reseal the ends so it becomes a circular thing again, and then you put it back into that bacterial cell and the bacteria is going to get busy making this protein. then you can just purify it at the end. So that's pretty commonly done these days. More advanced forms or newer forms of genetic engineering involve gene therapy, where you're actually inserting genes into human patients or modifying genes that are in human patients, instead of just taking a human gene and putting it in a bacteria and then having a bacteria make something for you. You're actually messing with the genes of the patient. So that's obviously a lot more risky and not, it's in its infancy, but it has a lot of promise for healing all types of disorders. This technology is all based on microbes. It's been researched in microbes, and microbes are used for it. So one cool example, well, cool or maybe scary, is this guy, Dr. Jiankui. I'm not sure how to pronounce his name actually, you guys. He actually took a gene, the specific gene, the CCR gene, and it's known that people who have HIV don't have this gene. Like, this gene confers resistance to HIV infection. So if you get HIV, then you don't have that gene. So he was able to take this gene and he actually inserted it into 2 newborn infants whose their father had HIV in China. This it was very recent, like a year ago. Yeah, I think like a year ago, maybe 2 years at the most. This was extremely controversial. So theoretically, like, when he puts this gene into these baby girls, they're going to be resistant to HIV. So that's right, good. And they had an HIV positive parent, but that doesn't really matter in this case because it was their father. HIV wouldn't be transmitted from a father to a child. It could be transmitted from a mother to a child, but not from a father, just because the father isn't the one who gets pregnant with the child. So they weren't necessarily really at like any increased risk of getting or being exposed to HIV, and he still did this modification, and I mean, since this is the first time that it happened, like, no one knows how safe it is. No one knows, like, what will happen to these girls as they get older. Hopefully everything's normal and nothing happens, and hopefully it all worked and it's great. But he didn't know whether it would work before he did this. So he's received a huge amount of criticism for that reason, and he's been stripped of his position, actually, his research position. Another more recent advance that was really important was direct observation of viruses. Viruses are a type of microbe that's extremely small, much, much smaller than any other microbe. And so you can't really see them with a light microscope. They're just too small. There's a couple of exceptions of ones that are big enough to see, but almost all of them are just way too small. They couldn't be seen until we invented the electron microscope, which was invented in 1932 by these guys, Max Knoll and Ernst Ruska. After it was developed, it took a lot of time to get the technology advanced enough to actually image biological sections. For one thing, biological specimens need to be sliced really thinly for the electron microscope to give good images. And the technology to slice tissues that thin was actually developed after the electron microscope was developed. So it took a while to get good biological images from an electron microscope, but gradually people did start imaging cells, which allowed them to see like structures inside the cell and allowed them to see viruses. So the first virus was actually observed directly in 1939 by this guy, G.A. Koch. And let's see, this picture here. This is the picture he took of the viruses. And it would be all these black blobs. Those are the actual viral particles. So you can see this is a pretty low quality image. The technology was not very advanced at that time, but he was able to see something. With advancements in the technology, we've gotten bigger pictures, better pictures. So this is a cluster of little viruses. And here you have a virus that's actually in the process of budding off of a host cell. So the virus is actually emerging from that host cell and it's about to like leave the host. So we also had bacteria and their effect on the environment being studied a lot in the 1900s. for the first time. This one guy, Martinus Beijerinck, was a very important scientist who made a lot of contributions to this field of microbial ecology. He studied soil and water microbes, and he found out kind of for the first time that soil and water is actually full of microbes. So no one really knew that before he did these experiments. And he found that it's hard to study these things because when you get a sample of the soil or the water, there's a bunch of different microbes in there, like all different types, all different species. If you want to actually study something, you need to get like one species alone so that you can see what it's like. So he was actually forced to develop or invent all these new culturing techniques to get that done, to help him isolate specific species from his samples. This is called enrichment culture. In enrichment culture, you're growing the bacteria in conditions that favor some species and are harsh for other species. So that might mean that you have like a temperature that's like just right for one guy, but it's too hot for another. Or it might mean that your media is like, it's missing something that like some species need and some species don't need it. So the species that don't need it are the ones that will grow. So basically just seeing how you can tweak those growing conditions so that some species are favored and other species aren't able to establish themselves in that culture. And if you do that, you start with your medium, your selective enrichment medium that has basically nutrients, but not all the nutrients, so that not just everything can grow in here. Then you introduce your sample. As you can see, it has all types of microbes in here. But the media, the enrichment media, it doesn't have everything that all of these guys need. Only some of them have everything that they need in this media. And in this case, it's these little red guys. So as time passes, those red guys, they have everything they need. They start dividing a lot. They fill up the media. And the other guys who don't have all the things they need, they're lucky if they can even just keep living. They can't really divide to make more of themselves. Then if you plate this, you're going to get colonies of just the red guy. So this is a way of isolating this one species from this complex sample with lots of species in it. Some of the important things that Martinus Beijerinck discovered would be bacteria that are able to use sulfur instead of oxygen, and bacteria that use other things instead of oxygen. He also discovered bacteria that can fix nitrogen by transforming atmospheric N2 into ammonia or nitrate. Those are really important bacteria in the environment. This another guy, Sergei Winogradsky, followed up on Martinus's experiments, studied those same soil and water bacteria, and he studied how they cycle sulfur and nitrogen through the environment. So he made a lot of contributions to our understanding of how microbes and bacteria in particular are important for cycling those nutrients from the environment and making sure that plants and animals have them available to them. One of the things that he did is make the term, or coin the term, lithotroph. Literally, lithotroph means stone eater. And he used this to describe bacteria that are able to use inorganic molecules for energy. Inorganic things like stone. So basically, bacteria that eat weird things that are not like organic, those are lithotrophs. So that includes a lot of the bacteria actually that are responsible for cycling sulfur and nitrogen through the environment, as it turns out. So microbes were also used to discover that DNA is the molecule of heredity, which was also in sort of the mid-1900s. So there were a couple of really famous experiments that are the basis for this discovery. First, you have Frederick Griffith's 1928 experiment, and then you have a follow-up experiment by Avery, MacLeod, and McCarty in 1944. So this first part of the diagram is Griffith's experiment. He took a specific bacterial species, Streptococcus pneumoniae, which is known to cause pneumonia. And he had realized that there's two different strains of Streptococcus pneumoniae, the R strain and the S strain. The R strain, when you culture it, it makes colonies that are rough, and that's why it's called R. The S strain, when you culture it, it makes colonies that are smooth, which is why it's called S. If you take a sample of this R strain and inject it into mice, the mice will live. When you take a sample of the S strain and inject it into mice, the mice die. So this experiment that he did shows you that the S strain is toxic, and the R strain is not toxic. He went further than that though. So he took a sample of the S strain and killed it. So he had the sample of dead S strain bacteria. He combined that with a sample of R strain that was living and plated that whole mixture, and he noticed that he got smooth colonies. So the only bacteria that were alive here were the R strain, which should make rough colonies and be safe, but he got smooth colonies, and then when he injected this into mice, he got dead mice. So that's weird. This is kind of showing him that something has gone from this S strain into the R strain. These dead S cells passed something on to the R cells that caused them to make a smooth colony and caused them to kill these mice. He didn't know what it was though, but this was demonstrating whatever it was, that's the molecule of heredity. And then the next step was just to figure out, what is it? So this follow-up experiment by Avery, MacLeod, and McCarty is addressing that question. So what they did is they took the S strain and they fragmented the cells and they separated the stuff inside the cells into DNA and protein. So they had a sample of S DNA and they combined that with living R cells. And when they plate that, they get smooth colonies. And when they inject it into mice, dead mice. It's not shown in this diagram, but they also did the control, not actually a control, but a different group where they had the S protein. They combined that with the R bacteria. Then when they cultured that, they got rough colonies, and when they injected it, they got living mice. So when you take the S strain is transferring something. Dead S cells are transferring something to R cells that makes them smooth and toxic. When you take just the protein from the S cells and put that with the R cells, they don't get anything. Like, they're still rough and they're still safe, non-toxic. But when you take the DNA from the S cells and put that with the R cells, then they become smooth in their appearance and they kill mice. So this follow-up experiment proved that DNA is the molecule of heredity. DNA is the molecule that's transferring information from the S strain to the R strain that makes that R strain toxic. Microbes and microbiology have been very important in developing the tree of life. So early versions of the tree of life were based on just what organisms look like, basically. If you look similar to something else, the scientists will class you as being like together or similar, and the tree of life is just organized based on the appearance of different organisms. And of course, it didn't really have microbes on it. There were some early trees that tried to incorporate microbes, but since it's all based on appearance, and the appearance of microbes is pretty simple, it wasn't really accurate. So an example of one of those early trees that tries to have bacteria in it is shown right here. This is like one of those kingdom-based trees where you have the plant kingdom, the fungus kingdom, and the animal kingdom. And at the bottom, Monera, that would be the bacteria. So you would have bacteria first, and then that branches out into protists, which we'll go over in a little bit, but they're really just simple eukaryotes, frequently unicellular eukaryotes that are frequently microbes. And then you have coming off of the protist or developing from the protist the plants, the fungi, and the animals. This is all based on just shapes. So it was really it was really difficult to put those bacteria into the tree of life in an accurate way because they don't leave a fossil, their shapes are very simple, and they don't really look like other living things. The only thing that they have in common with other living things for the most part would be like their basic structure and of their cells, and then the the genes, like the proteins, the genes, the actual molecules inside those cells. But you can't see that, even with a microscope, you can't see the molecules. So it took a lot more advanced techniques before you could start comparing those molecules to each other and classifying the bacteria more accurately. One guy whose work has been really important, or foundational actually, for building a modern tree of life is Carl Woese. He looked at the sequence of rRNA genes and used them to describe evolutionary patterns and relationships between all life forms, basically. So rRNA is ribosomal RNA. It's part of the ribosome that builds proteins. It's actually catalytic, so it's the part of the ribosome that actually links amino acids together to make a new protein. And rRNAs have rDNA genes. So most genes make a protein, but some genes make RNA, and some genes, rDNA genes, make rRNA for the ribosome. All life forms have ribosomes, all life forms have rRNA, and it always does the same thing. And it's very, very important. If you have a mutation in your rDNA genes, you're going to get different rRNA, and if it doesn't work right, you're dead. So it's very uncommon to have mutations in those genes that last and get passed on. So you can use those rDNA genes to investigate the relationships between things that had their last common ancestor a very long time ago, like people versus bacteria. Who could be further apart than that? But using rDNA, you can look at like how close we actually are to each other. So he took, let's see, here you have, well, I guess here's a diagram of rDNA and how that works. So here's your rDNA gene. This would be part of your actual chromosomes, or part of the genome for, a bacterial cell. And you're going to transcribe these genes to get pre-RNA. If you remember from general biology, that first you transcribe and get pre-mRNA, and then you have to process it. So after processing, in this case, you're getting mature rRNA, not mRNA, because this is rRNA. So now you have your rRNAs, and those are going to go into the different subunits of the ribosome. So when you look at this actual rDNA, you can see the sequence, and it's basically very close for different organisms, even if they're really far apart evolutionarily, because anytime something goes wrong here, you usually get a ribosome that doesn't work, and that means that that cell is going to die. So Carl Woese he sequenced the rDNA for all these different species, and he worked with a colleague, George Fox, to kind of analyze those different sequences and kind of categorize life based on similarities in those sequences. So they ended up finding 3 basic kind of patterns for rDNA sequences and divided life into 3 different domains on that basis. And the domains they made were Eukarya, bacteria, and archaea. And that's very recent. That was only published in 1977. So before 1977, no one really knew that archaea were significantly different from bacteria. They were just seen as a type of bacteria, but they're actually quite different. So that work has led to the modern tree of life, where you have the 3 domains, bacteria, archaea, and eukarya. This type of tree of life is a phylogenetic tree. What that means is that it's based on actual evolutionary relationships between organisms, and those relationships are determined based on genetics. In this case, genetics of rRNA. At the root of the tree, you have Luca, the last universal common ancestor. Then you have bacteria that have their bacterial rRNA sequences, archaea with archaeal rRNA sequences, and eukarya with eukaryotic rRNA sequences. The only thing that's missing from this tree is viruses. Viruses are not on the tree. Will they be one day? Do not know, actually. Theoretically, it's possible to put them on the tree of life because they have DNA. They have genomes. But in actuality, it's very difficult to do that because they don't have a lot of DNA. Most viruses do not have rRNA, for instance. And viruses actually seem to have arisen at different points. So not all viruses share one common ancestor, except for Luca. They have different ancestors, and they branched off of other life forms at different points. So you wouldn't have just like a virus domain. If they were incorporated into the tree of life, they would have little virus branches coming off of different parts of the tree. We're constantly discovering new microbes. Most microbes have never been described, discovered, or cultured in the lab. Woese and Fox actually were only working with cultured microbes. I mean, of course, those are easy to work with because you grow them in the lab and you always have samples available that you can study. But most microbes are not grown in the lab. They only exist out in the environment. So we barely have a taste of the true microbial diversity that's out there. And it's this guy, Norman Pace, who's done a lot to add to our understanding of how diverse microbes are. What he has done is take samples from the environment, like from the water, from the soil, even from like livestock farms and from like hospitals and stuff. And he just sequences that sample. Sequences the DNA from the sample, and based on that, he's going to get sequences for all the different species that happen to be in the sample, and he can kind of compare them and see. He can kind of make an estimate of how many different species are there, and sometimes find out something about their different characteristics. Like, well, for instance, he can find all different rRNA sequences in there from the different species, and that gives him an idea of how those different species that he sequenced would be fitting onto the tree of life. So using information from Norman Pace's experiments, we've been able to expand the tree of life for bacteria from this to this. So this would be the whole domain of bacteria. And each of these branches or little wedges coming off of it is a phylum. So under domains, phylum is the next biggest grouping of species. And based on Woese and Fox's data, they had figured out that there's this many different phyla. It's 11 different phyla of bacteria. Then when Norman Pace started adding in information that he got from his samples from the environment, he realized that there's many more phyla out there. Many more. So by 1998, he had expanded the number of phyla from 11 to 36, and now it's even bigger again. It just keeps going up. It just keeps going up as we find more bacteria. But there's so many of them. We'll probably never be done. And finally, we'll just go through the basic types of microbes, many of which we've already mentioned throughout the presentation. So you have microbes in all three domains of life. Bacteria, the bacteria are in domain bacteria. Then you have archaea. The archaea are in that domain. And then the rest of the microbes are in domain eukarya. So you have protists. Those are microbial. Algae, a lot of them are microbial. Some fungi are microbial. And even some animals are microbial. So if you look at this kind of diagram, it's showing you different microbes at different scales. So here's the smallest scale with the smallest microbes. This is a diatom, that's a type of protist. This paramecium, that's also a type of protist. And some of these, like yeast, that's a fungus. Some of these other guys are a bacteria. Here's cyanobacteria down here. Then if you zoom out, you can see other types of bacteria and archaea. This nanoarchaeum, that's an archaea. Archaea and bacteria are pretty similar when you just kind of look at them from the outside. They're little cells. When you look at the actual molecules inside of them, they start to look different. We'll get into that in a moment. As you keep zooming out, more bacteria, you actually start having viruses. These are some larger viruses. Influenza virus. This is a virus that only affects bacteria, actually, this T4 bacteriophage. And when you zoom out more, Actually, you can find a couple really huge viruses. So viruses are really small, except for like a couple who are really big. One of those here is the tobacco mosaic virus. It's like one of the giantest viruses ever, and it causes disease in causes disease in tobacco plants. Worms are not on here. So these tiny worms that are microbes, they're not actually mostly not microbes in their adult form. Like most of them are big enough to see with your eye when they're adults. Some of them are still too small to see when they're adults. You have to see them under the microscope. But even the ones that as an adult are big enough to see with your eyes are still classed by as microbes functionally in some cases because their eggs are microbial. Their eggs are always too small to see. And a lot of these worms are parasitic worms, and if you're going to kind of diagnose the person who has this infestation, you're going to be looking at eggs that they're shedding and their different wastes and stuff. So that since those eggs are microscopic, it has to be a microbiologist that looks at them. So for that reason, they'll frequently be considered in with the microbes. And apart from that, viruses. Right, so viruses do not belong to any domain of life. They've evolved at multiple points during the history of life. And are they even alive? You know, a lot of people think that they're not alive. Some people think that they are alive because they evolve. But in general, like they don't grow, they don't have a metabolism, they don't reproduce. So they don't do the things that we think of living things as doing, except for evolve. They have DNA, genetic material, and they evolve. Okay, so here's the bacteria. Bacteria are all unicellular prokaryotes, and they reproduce asexually. Their cell walls are made out of peptidoglycan. They live in almost every environment on Earth. They can eat all types of things. You know, any given species can eat, you know, only a given amount of things, but when you consider all the different bacterial species, they all eat different stuff. So as a whole, they can eat just about anything. They're extremely diverse. Now we have over 80 phyla of bacteria. In 1998, it was 36. In 1977, it was 11. In 1998, it was 36. And today, in 2020, it's over 80. A few of those cause disease. A few of them actually protect your health by being a beneficial part of your microbiome. And they're also extremely useful for technology and for, I guess, bioremediation, for getting rid of pollutants and wastes. So these are some different shapes that bacteria can have. Of course, little circles, little rods. They can also have like weird shapes, like they can be spirals. They can look like kidney beans and just have I don't know, shapes that are hard to find a word to describe. Weird shapes. Next you have archaea, which are in their own domain. Those are also unicellular prokaryotes that reproduce asexually. They do not have peptidoglycan in their cell walls. So one of the easy ways to differentiate them from bacteria would be based on their cell wall. They don't have peptidoglycan in it. They have different things. They also live in all the environments of the Earth, but we frequently think of them as living in extreme environments. Not all of them do. A lot of them just live in regular old places. But a lot of extremophiles are archaea also. They also can eat a wide variety of things and weird things like metals and stuff. And if you look at them genetically, they are different from bacteria. They have different rRNA. They also have other molecules that are different, like their cell walls are different, and a lot of things inside them are different from bacteria that you can't just see by looking. And archaea are not known to cause disease. So actually we don't know of a single disease that's caused by an archaea. Here's some drawings of different archaea. As you can see, it's again balls, rods, weird stuff. So they look, when you just look at them in the microscope, they look real similar to bacteria. Actually, you have to kind of analyze them a little closer to notice the differences. Next, you have protists. Protists are a type of eukaryote. Another word for protist is protozoan, so protozoa would be the plural. Protist is kind of a weird grouping. It's not really based on genetics. It's kind of a catch-all term for just any unicellular eukaryote that's not an animal, plant, or a fungus. So it's like a grab bag, basically. So if there's a eukaryote, but it's not an animal, and it's not a plant, and it's not a fungus, you basically say it's a protist if you don't know where else to categorize it. Most of them are unicellular, but there actually are a few that are multicellular, mostly unicellular though. So since it's like a grab bag group, they don't have they're not all closely related to each other genetically. They don't belong to a single cohesive group based on genetics or phylogenetics. So it's a strange grouping that as we add more details to the tree of life, we might at some point stop using because it's not based on genetics, and it's not based on evolutionary relationships. Most protists are asexual, and they mostly live in the water. So they can live in different places, but mostly in the water, where they form the basis for marine food chains, like a plankton. A lot of plankton are protists, and whales eat them and stuff. Most of them don't cause disease, but there are a few of them that do cause disease. So this is an image of different different protists living in water. As you can see, they have really crazy shapes to them, and they're very diverse. Next, you have algae, which are also eukaryotes. It's another grab bag kind of classification. So in order to be an alga, you have to be a eukaryote who does photosynthesis and is simple, so not a plant, simpler than a plant. So a lot of different things qualify as algae. They all do photosynthesis and have different simple structures. So that's not a genetically cohesive group, which means the algae are not all closely related to each other by evolution. And again, at some point, it's possible this grouping could not be used anymore if it becomes more obsolete. You actually do have some algae that don't do photosynthesis anymore because they've lost it. They've just lost that ability, but they still have some of the molecules that they would need for photosynthesis, so you can tell that they used to have it. Algae are pretty diverse. They can be multicellular or unicellular. Most of them reproduce asexually, but some of them reproduce sexually. And some of them are not microbes, some of them are. So these are examples of different algae. These would be all microbial except for this one, this alga one, that's seaweed. So kelps and seaweed, those are all types of algae. Those are multicellular algae species. Those are not microbes, of course, they're big. And they almost all live in the water, where they also, like the protists, help form the basis of aquatic food chains. Algae are really important for the environment because since they do photosynthesis, they make oxygen. So they help oxygenate the atmosphere and maintain the level of oxygen on our planet. They do not cause disease. There's not even one alga that causes disease. They are very useful though in technology. So Jello, that that comes from algae, and agar for the lab for growing bacteria, that's also coming from algae, and other things as well. Then you have fungi. Most fungi are not microorganisms, but some of them are. They are all eukaryotes. Most of them are multicellular, but some of them are unicellular. Some of them can actually be multicellular or unicellular, weirdly. They're weird. And they can also reproduce asexually or sexually. So a lot, some of them can only do one or the other, but most of them can do both actually. They do have cell walls. Some of them cause disease. It's not very common. There's not a lot of diseases that are caused by fungi, but some of them do cause disease. A lot of them are useful in bioremediation to recycle organic waste. In terms of the fungi that are microbes, You have two major types, either yeasts or molds. Molds are multicellular. They reproduce asexually and sexually. Yeasts are unicellular and they reproduce asexually. And you have a lot of species that can either make a mold form or a yeast form, so they can be both. They can differentiate. So here you have an image of yeast cells. Here's an image of a mold. This is a reproductive structure on a mold. So this mold is microbial, but it's made out of multiple cells. And this would be a structure for sexual reproduction that is made here. These yeast cells are single-celled. The little dots that you see are new yeast cells that are budding off of the old ones. So that's asexual reproduction that you're seeing. And these are all different types of fungus that have both a mold form and a yeast form. So depending on what conditions you grow them at, they'll either make a mold or a yeast, which makes it pretty hard to classify fungi sometimes. And then microscopic worms. Most of these are harmless. An example is C. elegans. That's not only harmless, it's beneficial because because we use it as a model organism in science a lot. So just an organism that is frequently studied and used as a subject for experiments. So this here is a C. elegans worm. It's microscopic from the day it's born till the day it dies. It's real tiny. And you also have some worms, some of these microscopic worms that are parasitic. So some of those will stay small their whole life. Some of them are going to start as microscopic eggs and then become something bigger like a tapeworm. Finally, viruses. Viruses are weird because they don't have cells. They are acellular. So not unicellular, not multicellular, acellular. And you know, they may or may not even be alive. So they don't have metabolism, they can't reproduce. Those would be kind of the main arguments for why they're not alive. But they do evolve, and that's the main argument for why they are alive. Viruses are always parasites of one sort or another, so not all of them cause disease in people, but all of them are parasitizing something. Since they can't make their own energy in metabolism and they can't reproduce themselves, they have to steal everything they need from a host. They have to force a host to make energy for them and to build new viral particles. You have some viruses that are real harmless actually. They don't do anything bad to you. They just kind of live in your cells. They are very well adapted to the host, so they don't hurt the host. It's actually better for the virus to not hurt the host because if the host dies, it doesn't make any more virus. But a lot of viruses are not that well adapted to their host and they cause disease. You actually have a handful of viruses that can be useful. So an example would be the European chestnut is a tree. Well, okay, the chestnut tree. There's several different varieties of chestnut trees. You have the European chestnut, American chestnut, and Japanese chestnut. Chestnuts used to be real common in America. It was considered very lucky to have a chestnut tree growing on your property because they make chestnuts, a lot of them, and they're, yummy for eating. It's like free food. But then you started having this fungus going around, the chestnut blight fungus, that killed basically all of the American chestnuts. You don't see those trees anymore. The fungus is a weird fungus. It doesn't hurt the tree when it's a baby, but it hurts it when it's an adult, before it's going to bear chestnuts. So you can see like baby American chestnuts, but they don't grow into a full tree. And there's scientists who are trying to figure out like how can you save and bring back the American chestnut and save it from this fungus. We don't have a cure for the fungus, but what we have found actually is that there's some viruses that will infect that fungus and make it less virulent. So that means make it less able to cause disease. So if you give the chestnut blight fungus a virus, then maybe it won't be so harmful to the American chestnuts. So we haven't, I guess, developed that strategy to the point of actually deploying it because, what if something goes wrong and things end up worse than they are now? But it's possible that might work out in the future to bring back chestnuts in America. Right, the European chestnut is where those viruses were discovered. The European chestnut doesn't suffer from this blight, and it's because the chestnut blight fungus in Europe has these viruses infecting it that make it less virulent. Another example of a virus that's helpful would be like the adenovirus, which is actually pictured right here. The adenovirus, actually a lot of different viruses are adenoviruses. That's a type of virus actually. And some of them cause a common cold, so they can be, bad for you. But they've also been used in viral gene delivery technologies. So because of the viral life cycle, which we'll get into in more detail later in the semester, viruses are designed to put their DNA into a host cell. So that means that you can theoretically use them to deliver DNA to cells. Cells don't normally take up DNA. Like your cells, if there's some DNA lying around, they're not interested in it. They don't want it. They're not going to take it up. So if you're doing gene therapy, you have to not only inject a person with this modified DNA that's going to help them, you have to inject it in a format that is actually going to go into their cells, and it won't normally. But if you put that DNA, that modified therapeutic DNA, into a virus, like a modified adenovirus, you take out the adenovirus's DNA and you put in your therapeutic DNA, and then you inject that into a person, then the adenovirus will deliver the DNA into their cells, but it won't make them sick because it doesn't have its own genes anymore. So there's a couple different viruses that are modified and used as gene delivery vectors.